Today we're covering an intracellular process known as substrate-level phosphorylation, including when it occurs and how it differs from oxidative phosphorylation. We know that the human body derives energy from food, but to use food's energy, the body's cells must first break down food's molecules into adenosine triphosphate, or ATP, the primary energy currency of cells. This process is called cellular respiration. When energy is required, ATP's high-energy phosphate-to-phosphate bonds can be broken to release energy. Substrate-level phosphorylation is one crucial mechanism by which cells generate ATP. Let's take a closer look. Phosphorylation is the process of transferring a phosphate group from any donor molecule to any acceptor molecule. Substrate-level phosphorylation is a kind of phosphorylation that refers to transferring a phosphate group directly from a phosphorylated intermediate, known as the substrate, to adenosine diphosphate, ADP. This results in ATP and another product. Kinases catalyze the reaction. Substrate-level phosphorylation occurs twice during cellular respiration. Once in the cytoplasm during glycolysis, the first step of cellular respiration, and once in the mitochondria during the Krebs cycle. It's often represented with a curly arrow like this, from ADP to ATP. Let's look at the first instance during glycolysis. Can you spot the substrate-level phosphorylation steps? 2 ATP molecules are gained along the way by substrate-level phosphorylation. As 1,3-bisphosphoglycerate, the substance, is converted to 3-phosphoglycerate, the product, and then again when phospho-enol pyruvate is converted to pyruvate. What about during the Krebs cycle? Substrate-level phosphorylation occurs as the enzyme succinyl-CoA synthetase converts succinyl-CoA, the substrate, to succinate, the product. Substrate-level phosphorylation is not to be confused with oxidative phosphorylation, a distinct, more complex process that couples phosphorylation with oxidation. Unlike substrate-level phosphorylation, oxidative phosphorylation relies on energy derived from the electron transport chain. Compared to oxidative phosphorylation in the mitochondria, substrate-level phosphorylation is a relatively minor contributor to the ATP in the cell. However, it's still necessary. It is a quicker ATP-producing process than oxidative phosphorylation, and it can produce ATP independently of oxygen. This becomes especially important in anaerobic conditions, like in muscle cells during intense exercise. In this lesson, we've covered substrate-level phosphorylation, when and where it occurs, and how it differs from oxidative phosphorylation.